So let's go ahead and take a look at how we go about forming urine through the interaction of the nephron with the surrounding capillaries and arterioles, so as to create either a very concentrated urine or a very dilute urine. So the principal mechanism that's going to come into play here is the countercurrent multiplier. And what the countercurrent multiplier is going to allow for is it's going to allow for an interaction between the nephron and the surrounding capillaries and arterioles and venules, so as to allow for either a very concentrated urine or a very dilute urine to be formed based off of the osmotic conditions of the surrounding tubules, where the end result is going to be a filtrate that is around 4 to 4.5 times as concentrated as the initial filtrate when it first entered into the nephrons. So let's take a look at this process.
The kidney is the body's filtration system and produces urine in its thousands of nephrons, the kidney's basic unit. Each nephron begins with the Bowman's capsule, wrapped around a capillary ball, the glomerulus, which becomes a tubule that eventually forms the loop of Henle, before collecting into tubules and ducts that empty into the bladder. Fluid from the blood enters the nephron by filtration and passes along it, where substances are added from the blood by secretion or removed from it by reabsorption. Potassium and hydrogen ions, organic acids and bases, metabolites and foreign substances are secreted across a selective membrane. The potassium and hydrogen are exchanged for sodium along the tubule. 99% of the filtrate is reabsorbed through a mixture of passive diffusion down an osmotic or concentration gradient and active transport by pumping against a concentration gradient. The kidneys can conserve water. The only way that the body can move water is by moving sodium chloride first. You have to create a concentration gradient so that water can move by osmosis in the direction of the highest concentration. The loop of Henle creates a hyperosmotic region in the medulla of the kidney, acting as a sponge, drawing water out from the collecting duct, if the duct becomes permeable under the influence of vasopressin, also known as antidiuretic hormone or ADH. The ascending limb of the loop of Henle, which is impermeable to water, pumps out sodium chloride via sodium-potassium chloride co-transporters from the urine into the blood in the vasa recta. Blood in the vasa recta carries this sodium chloride into the medulla, making the entire region hyperosmotic. The blood, which is moving in the opposite direction to the urine in the loop of Henle, then ascends parallel to the descending limb of the loop of Henle. This descending limb is water permeable, allowing water to move out of it into the vasa recta. This entry of water into the ascending vasa recta reduces the osmotic potential of the blood to normal as it returns to the body. The interplay between the loop of Henle and the vasa recta is called the countercurrent multiplier. Essentially, the medulla acts as a sponge, retaining and releasing water to address the water requirements of the body. ADH acts on the collecting duct to make it permeable to water, allowing water to move into the hyperosmotic medulla, where it is drawn up by the vasa recta and returned to the body.
So let's take a look at this interaction as it relates to the movement of solutes in terms of creating that hyperosmotic isoosmotic and hypoosmotic conditions. So we first start off with movement of fluids from the plasma into the nephron, generating an osmotic condition that is isoosmotic to the plasma. We will then have exchange of solutes as well as extraction of water from the proximal convoluted tubules as well as from the descending limb of the loop of Henle. As we exit the descending limb, we are unable to have water movement. So the only ability to have a concentration or a dilution of the filtrate is through active pumping of ions, in particular sodium and chloride, through the ascending limb. So if we notice, as we come down the descending limb, we get more concentrated, the numbers increase. As we head up the ascending limb, we become more dilute, we get a reduction in the concentration of the filtrate, the numbers get smaller. As we head into the DCT, the distal convoluted tubules, we actually get to a hypoosmotic region. We then start to have exchanging of some solutes, in particular hydrogen ions and potassium ions within the distal convoluted tubules. We will have some movement of water in accordance with that. Within the collecting tubules, we'll start to have a cycling of urea. As we start having a cycling of urea, we will start to have more reabsorption of water, and we start to concentrate the filtrate once more. As we exit from the collecting tubule and head to the collecting duct, the principal cells, due to ADH and aldosterone expression and responsiveness, will allow for additional reabsorption of water, which allows for the final concentrating of the filtrate. As we exit the collecting tubules and enter into the collecting duct, as we head through the papillary membrane, we have now formed urine. This whole sequence of events is the formation of urine. So what are the consequences of this countercurrent multiplication system? We'll be able to collect about 8 liters per day of whatever fluid was left following the proximal convoluted tubules reabsorption. We'll be able to collect about 2/3 of all sodium chloride. We'll be able to generate osmolarity conditions within and surrounding the nephrons so that we are able to generate a concentration of urine that is between 12 and 1400 milliosmoles, somewhere between 3 and 4 times, and sometimes up to 5 times as concentrated as the plasma that as it first entered into the filtrate. Now, this number in terms of milliosmoles is going to be dependent upon the concentration of the plasma itself, which means that if someone is dehydrated, their actual milliosmol concentration of filtrate will change accordingly. So if we start, so we're basing this off of a 300 milliosmol, if all of a sudden the person is a 400 milliosmol, instead of going from 12 to 1400, we're now going to go from 1600 to 2000 milliosmol. And so that's one of the things that happens in terms of the consequences of dehydration in regards to this consequence of countercurrent multiplication. The other thing that comes about from this is with expression of loop diuretics is that instead of generating a 1200 milliosmol concentration at the distal end of the descending limb, we're only at a 900 level or a 700 level. And what this does is this ensures that we are able to remove more fluid than what we would have otherwise. So the entirety of all this processing is the formation of urine. And so the formation of urine ends once we have exited the collecting tubules and entered into the collecting ducts. We say we formed urine at that point in time because we have no other means to go about recollecting any of the filtrate that may have been passed along. The process begins with glomerular filtration. We then have all of the exchanges that take place, the secretions and reabsorptions through the nephron, the countercurrent multiplication, with the final terminal product being formed as we exit into the collecting ducts. So what is the chemical composition of urine? There's a much more detailed table within the study packet And we'll get more detail when we talk about urinalysis and what we're actually measuring within the urine during urinalysis. Regardless of the volume that's being produced, 95 to 96% will always be water. We measure the inorganic and organic molecules based off of two factors. The principal factor we use to measure this is often referred to as the milli equivalent. That is the indication of how much water drawing property those molecules have. And once again, we'll get more detail into this when we look at urinalysis. The principal organic molecules that we're looking at here are the primary metabolic wastes, which include urea, nitrated urea, creatinine, do not confuse this with creatine, uric acid, and then we have metabolic acids that would otherwise be some sort of toxin to normal metabolic processes. In the special considerations, there is a summary about the detoxification principles that take place within the liver and within the kidney. Remember that your liver, your kidney, and your lungs are the principal detoxifying agents within your body. And regardless of what people are trying to sell you, there is no such thing as a detoxification agent that you can purchase and utilize. It's what your body naturally is doing. There's a whole bunch of reactions that we looked at within the liver that's going to be principally doing most of this detoxification, but we have very similar reactions within the liver happening within the kidneys that allow for the same type of detoxification to take place. Now, there's one interesting feature that we have to remember about in terms of the volume of urine that's being produced, is that the volume of urine is going to be directly regulated by how much sodium ions we allow to go into the excrement, to go into the urine. Now, sodium is not a waste product, but what it is, it's the regulation of how much water we're being allowed to lose. And it goes back to that same principle they always talked about in terms of water following salt and salt following water.